when biology learned to edit itself. It started with a cut so small, it was almost invisible. A single change inside a living cell. And yet, that tiny alteration carried the power to rewrite life itself. To understand why this matters now, we need to look at how biology once worked. For most of human history, life followed fixed instructions. DNA was destiny. Traits passed forward, mistakes accumulated, and evolution moved at a pace measured in generations. Humans could study biology, treat its failures, but never truly change its code. Until that assumption quietly collapsed. The pressure came from limits we could no longer ignore. Genetic diseases without cures, crops failing under a warming climate, viruses evolving faster than medicine. Biology was powerful, but slow, and the modern world does not wait. So scientists began searching for a way to intervene directly, not by adding chemicals or performing surgery, but by correcting the source itself. The breakthrough arrived from an unlikely place. A bacterial defense system, evolved to fight viruses, revealed a method of precision editing. It could locate a specific sequence of DNA, cut it cleanly, and allow new instructions to take its place. At first, it seemed like a clever laboratory trick, a useful tool for research. Nothing more. But then experiments accelerated. Cells were edited, and then organisms. Errors vanished. Traits appeared. Diseases reversed themselves at the molecular level. Biology was no longer something we observed from the outside. It was something we could revise from within. The implications spread quickly. If life's code could be edited like text, then limits once considered permanent became temporary. Inheritance became adjustable. Evolution became intentional. And humanity stood at the edge of a question it had never faced before. When biology can edit itself, who decides what should be written next? At first, the changes felt contained. Edits were small, targeted, and therapeutic. A faulty gene corrected. A missing protein restored. Patients improved in ways medicine had never achieved before. What once required lifelong treatment could now be resolved at the source. But the technology did not stay confined to hospitals. Researchers realized that the same tools could enhance resilience, not just repair damage. Crops were engineered to survive drought. Livestock resisted disease. Cells were modified to produce materials, fuels, and medicines inside their own biology. Life became programmable. With each success, the boundary between treatment and enhancement blurred. If a gene edit could prevent illness, could it also increase strength? If it could correct vision, could it sharpen it beyond human norms? Biology had never asked permission before. Now it required choices. The conflict deepened when edits became heritable. Changes no longer ended with one body. They carried forward, shaping generations yet unborn. A decision made in a laboratory today could echo for centuries. And once released into populations, genetic changes could not be recalled. Supporters argued this was the next step in human responsibility. If suffering could be removed at its source, refusing to act became a moral failure. Critics warned of unintended consequences. Ecosystems are fragile. Genes interact in ways no model fully captures. A single change could ripple outward, reshaping life in unpredictable ways. Then came the first real controversy. An edit performed not to heal, but to improve. The results were ambiguous. The science worked. The ethics fractured. Some saw progress. Others saw a line crossed without consent. And beneath every debate sat an uncomfortable truth. Biology was no longer guided only by natural selection. It was guided by human intention. Once life becomes editable, restraint becomes the hardest discipline of all. As editing tools spread, the consequences moved faster than the rules meant to contain them. Laboratories around the world gained access to techniques once limited to elite institutions. Costs dropped, barriers fell. What began as a guarded scientific capability edged toward accessibility. This was where the tension truly surfaced. Biology does not operate in isolation. Edited organisms interact, reproduce, and compete. A crop engineered for efficiency could displace native species. A resistant insect could alter an entire ecosystem. The edits were precise, but the outcomes were not. Inside human biology, the stakes rose even higher. Editing cells inside a living person carried risks that only time could reveal. Off-target changes, long-term effects, interactions no one anticipated. The edits solved one problem while quietly introducing others. Yet the momentum did not slow. Success stories multiplied. Children spared inherited disease. Treatments that worked where nothing else had. The benefits were real, immediate, and deeply human. Each success made hesitation harder to justify. Then came the deeper shift. Biology was no longer only reactive. It became predictive. Scientists began designing organisms for futures that had not arrived yet. Climate-resilient plants for environments decades away. Immune systems trained against viruses that did not yet exist. Life was being shaped in anticipation of tomorrow. This raised a haunting paradox. By editing biology to survive future threats, humanity was also narrowing nature's ability to surprise and adapt on its own. Diversity gave way to optimization. Flexibility surrendered to efficiency. The conflict reached its emotional peak when researchers realized something irreversible. There would be no return to untouched biology. Even choosing not to edit was a decision with consequences. Inaction allowed suffering to persist. Action risked altering life forever. Biology had always evolved through chance and pressure. Now it evolved through choice. And humanity was still learning how heavy that responsibility truly was. The moment of reckoning arrived not through disaster, but through clarity. Biology had crossed a threshold where intention mattered as much as discovery. Editing life was no longer a question of capability. It was a question of values. Scientists began to slow down. Frameworks emerged. Global conversations took shape around consent limits and shared responsibility. The focus shifted from what could be edited to what should be protected. Diversity, uncertainty, the right of future generations to inherit choices they did not make. In medicine, gene editing settled into a careful balance, powerful but constrained, life-saving but monitored. In agriculture and ecology, the emphasis turned toward coexistence rather than domination, editing not to replace nature, but to support it under pressures humans had created. Yet the deeper change was philosophical. Humanity had always adapted to biology. Now biology was adapting to humanity. Life was no longer just inherited. It was curated, and with that came a new understanding of stewardship. Editing biology did not make humans gods. It made them caretakers of a system more complex than any code they could write. Every edit carried trade-offs. Every solution carried new questions. Progress demanded humility. What biology taught us in this moment was simple and unsettling. Power without wisdom accelerates risk, but wisdom without courage leaves suffering untouched. The science that learned to edit life did not just rewrite genes. It rewrote humanity's relationship with nature itself. Sometimes the smallest shift in perspective becomes the beginning of a new future.